I will talk about the lower motor neuron types of injury when it comes to spinal cord. And this is something that we have been interested in our lab for quite a while now in focusing on these types of injuries that for a long time have almost been forgotten by the research community. When we talk about the Conus Medullaris and Cauda Equina forms of injury, what we really mean are injuries that take place at the lowermost portion of the spinal cord, the sacral portion of the spinal cord, and also the nerve roots that extend from this area and that are called the Cauda Equina. This means the horse's tail, and as you can see how these nerve roots together create that shape here. And when trauma hits this area, there is a special form of spinal cord injury that develops that is quite different from injuries that would affect the neck or the thoracic region. When we talk about levels of injury, these are injuries that typically would affect around the lowermost thoracic vertebrae like T12, L1, L2, and that would affect the conus area, which is the sacral spinal cord, and these nerve roots in this area. And when these are anatomically complete, what it leads to is a different kind of weakness. It's not a spasticity, it's more of a flaccid weakness. It's more like a ragdoll type of weakness in the lower extremities. So the functions that are affected with these injuries are especially the lower extremities, leg weakness, numbness of the lower part of the body, and also bladder, bowel, and sexual functions are also affected by these injuries. And in addition to the absence of function, there is commonly also development of pain after injuries to these regions. So I will be talking about how we have approached this from the research perspective and how we are trying to both understand the injury itself, but also how we can hopefully turn things around and come up with new translational approaches for repair and treatment. So this is a schematic of the lower urinary tract function in rats. And the big principles are similar in all mammals, also in humans and other mammals. And what I wanted to point out here is that there are two important components. One is a component of autonomic neurons, meaning neurons that are under non-willful control that innervate different ganglia and the visceral organ here, the bladder. But we also have motor neurons that innervate skeletal muscle in urethra sphincters. And those two components are very, very important for the normal function. And when we have a spinal cord injury that would be a high injury, let's say at the cervical or thoracic level, there's a disconnection syndrome affecting these neurons, meaning that they lose contact with the brain and the brainstem and so forth. But when we have an injury to the lumbosacral level or the conus, you have an injury directly to these nerve cells. And this is what happens in the conus injury and also at the Cauda Equina injury. It's a direct hit at these neurons that innervate the visceral organs and motor neurons, not only of sphincters, but also of the extremities. When we started looking at experimental studies on this, we were interested in developing research models that would mimic the human condition. And we were looking at this as a lower motor neuron problem, just like what Donna was mentioning here, the lower motor neuron that creates the denervation of the peripheral organs. In many of the spinal cord injuries, you have a a denervation of the part of the spinal cord below the level of injury. But here we have a denervation of the peripheral target organ, such as a muscle or a bladder. And so what we did in our early work was to do a nerve root lesion of the ventral root that carry the motor fibers, but also the autonomic fibers, that we avulsed these roots from the surface of the spinal cord to create a disconnection with the periphery. And what we found was that this kind of injury leads to a lot of changes inside the spinal cord, but also includes loss of both motor neurons and the autonomic neurons. So this injury is actually lethal to nerve cells, and it's also lethal over time. So there's a time course here. That means that time equals neurons. So the longer out you're in this time course, the more neurons you're losing after the injury, which also creates an opportunity for treatment, because if you can go in early and treat, just the way we think about maybe strokes and heart attacks with early treatments, that spinal cord injury may also be something that we should be maybe more aware of the clock ticking after the injury. And at least the experimental work here indicate that is the case for both autonomic and motor neurons. What we did here was to, as an initial attempt of treatment, was to surgically reconnect some of the ventral roots that were separated from the spinal cord. And we were inspired at the time by some pioneering work that Dr. Thomas Karlstedt had been performing both at the Karolinska in Sweden, but also in London, where he had studied both experimental models and also the first human cases by implanting nerve roots at the level of the neck after brachial plexus injuries. And he had seen some early motor reinnervation of at least some proximal peripheral targets. So we were also intrigued by this to see could we do something similar but looking more at autonomic function. So that was our background to this. And to do this in a stepwise manner, we went from the injury to the initial repair strategy, and we found that from the get-go, we were able to more than double our surviving neurons. So these roots, when they are repaired and surgically replanted into the spinal cord, have a protective effect. We were also able to show that when we look inside of the roots, there are a lot of axons growing. Up here to the top left, you will see a control nerve root. In the middle here, you see the atrophied root after injury, and to the right here, you have a root after repair. And even though it doesn't look fully normal, if you look at the higher magnification, you will see there are numerous fibers growing, and in numbers that are very close to the normal. And actually, there are in that image there. When we look in the electron microscope, we find that there are Schwann cells, which are the myelin-producing cells, creating these sheets around the regenerating axons as well. So it was really a good sign that we were able to bring the axons growing from the spinal cord to the periphery. And that is a very, very important hurdle, because if we cannot achieve that, we are stuck. So that was a very, very important and critical experiment for us in this development. We also looked a bit more in detail at the avulsed root. So here again is a normal control ventral root. Here is a root that is undergoing what is called Wallerian degeneration. There was a scientist called Waller who gave the name Wallerian degeneration of these degenerating roots. And when you look at this, it looks like everything here is dead and gone. But when you bring the sample into the electron microscope, you will see at the bottom here that there are cells that are alive, and many of them are Schwann cells. And not only are they alive, they are very metabolically active, and they even go through cell divisions and multiply. And when we work with functional genomics colleagues at UCLA and look at what these cells are producing, it turns out that they are secreting and producing a lot of growth factors. And this is probably why we are able to attract the axons from the spinal cord out to the periphery, that the growth factors are released by these cells and attract the growing axons to the periphery. So we have recently developed some techniques in collaboration with Dr. Ahmed Hoke, a neurologist at Johns Hopkins, and an engineer at Johns Hopkins called Hai-Quan Mao, where they produce nerve guidance tubes of biodegradable materials that we can actually put different growth factors inside. And in our gene expression studies, we have seen a variety of growth factors, and one of them is a growth factor called GDNF, which is normally very important for motor neurons. And when we look at GDNF in these studies, we're not only able to see that GDNF is secreted, but also in these types of network analyses, we can also see how GDNF is connected to other genes that are regulated inside of these injured roots. And so this is just a proof of principle that we can take this approach and study the effect of the growth factor, and in this case see that we get a very prominent growth of axons compared to empty tubes with also myelination. As you can see on the lower end here, these kind of dark circles around the axons here are the myelin, and these are really stimulating the growth. So this is kind of part of the mechanism as we believe, and that is all good. But so far, all we've done is to accomplish. We have cell protection, and we can get axons to grow to the periphery, but we have no function yet. So what we did next was to expand our injury model to a bilateral injury model. So in this case, we avulsed the roots at multiple levels and surgically replanted on both sides of the spinal cord in this fashion to get a surgical reconnection. You can see that here. These are surgically reattached roots to the spinal cord. And so what we're hoping is that we can have those roots support the growth of axons for an extended period of time. And what we were targeting, because we are working in the Conus Cauda Equina territory, is to see, can we get function back of bladder control? So in order to do that, we needed to develop a way of analyzing this. And you will see here the setup of what's called a urodynamic study, which essentially is a catheter that is placed into the bladder to measure the pressure that we can read on a computer. We can also record with EMG electrodes from the sphincter muscle at the urethra and record this in real time. And when you infuse into the bladder will expand to a point and then contract as a reflex. And these are traces that we can record from and analyze how well a bladder is doing this. And so these are traces from rats, where we can see both the bladder pressure changes and the EMG changes from that take place during a normal filling and voiding. And this is really the same setup as you or I would have if we go to a urologist for a bladder evaluation. The only difference would be that we would probably not get the sedation that the animal will get, but because this is an outpatient procedure in humans that is done mostly with us being awake. So it's a nice outcome measure because it's clinically relevant. It's something that is used in the clinic that we're also now using in the laboratory, so that if we see changes in the laboratory, we also then know what to look for in the patient population when treatments are being evaluated. So what we see here is to the left in the upper portion are normal traces from bladder contractions, and the lower part here is EMGs, and these are synchronized and normal contraction cycles in the rat undergoing a urodynamic examination. This is your normal, but this portion here with a flat line here, that is after a bilateral injury to the Cauda Equina, when we have avulsed bilateral lumbosacral roots and disconnected the spinal cord from the visceral organs. So in that setting, because we have a disconnect between the spinal cord and the bladder is unable to respond when we fill it with saline, because it doesn't have the nerve connections to cause the contraction. So it is showing up as a flat line on the traces here. So we can just fill but never get a contraction. But with our approach of surgically repairing the injured nerve roots and replanting them back into the spinal cord, we waited for about 3 months, 12 weeks, and we were very, very excited to see that the animals were showing recovery of reflex function. So you see these traces here, both this one and that one, meaning this one and that one, they show return of bladder contractions and also activation of EMG activity. That is also appears to be somewhat synchronized with the bladder contraction. And so even though it doesn't look normal altogether like this, it is a lot better than having a flat line and no connection whatsoever. So this is really the return of reflex micturition with this approach. To the right side of the slide, you see some sections of the spinal cord where we have done nerve cell counts and also labeled nerve cells in the spinal cord from the periphery to show that it's not just a functional recovery. We also have an anatomical reconnection between the periphery and the spinal cord. We have injected tracers into the sphincter muscle that is picked up and transported back into the spinal cord. So we have also the anatomical connection there to back up a bit more the functional recovery. And also here, we were able to show that the survival rate is increased by this procedure, and the more nerve cells that survive, the better the function. So that comes back to the issue of timing of repair, and that early repair is likely to be more efficient than late repair, because you want to rescue as many neurons as possible so that you can get as good of a function coming back as we can possibly have. So being encouraged by these findings, we also wanted to bring this a bit closer to a human intervention. And since we have a surgical procedure that also inflicts an injury to the spinal cord, one of the concerns one could have is the potential development of pain. And so what we did to address this issue early on is to develop a model where we assess pain in these animals. So again, these are rat studies, and we do the avulsion of the ventral root. You see the lesion here at the surface of the spinal cord there. We keep the sensory fibers intact, but we just do the lesion of the motor fibers here. And what we were concerned about was when we replant these roots back into the cord and then increase the injury, could that cause pain? But in our experiment here, we found that when we do the injury only, we do develop a pain called allodynia. And allodynia is when you have a stimulus such as light touch, or something that normally is not painful, but becomes painful as an abnormality. And this is not uncommon in spinal cord injury or other traumas to the nervous system, where, let's say, the clothing could be uncomfortable, it could be over the level of the injury, like an at level pain syndrome, like a band around the area, or it could even be below the level of injury in humans. It could be the wind blowing, it could be the sheets that touch your legs, or the stomach when you're trying to sleep. It could be all those kinds of things that are examples of allodynia. And so what we were surprised to see here, that even though we did not do a sensory injury, we still had a sensory consequence. That was a bit of a surprise to us. But the second part of this was when we surgically replanted these roots back into the spinal cord, that initial pain gradually disappeared, and the sensory levels came back to baseline. And what we were not aware of at that time was that Dr. Karlstedt, who had operated some of the patients with brachial plexus injuries, actually had published clinical cases where he reports that patients are coming in describing some recovery of proximal function, but also improvement of pain. And so we had a very interesting conversation about this that also resulted in a book chapter that we wrote together about sensory consequences of root injuries, and how little we really know about the mechanisms, but it was kind of interesting that in this case that the first examples came from the clinical literature, and then we serendipitously were able to demonstrate that there is also a model to study this in rodents. These slides are just to show that in the spinal cord we also get aggregation of microglial cells, which are a kind of inflammatory cell in regions that would be expected to be affected in allodynia situations. So it's not diffuse in the spinal cord, but certain parts of the dorsal or the sensory areas of the spinal cord. Similarly, some of the nerve chemicals or transmitters are also affected in select regions. Here is a glutamate marker that we have a downregulation for in the dorsal horn, and then it's coming back after repair. And I should also say for the microglia marker, it also was restored towards normal after the repair site. So it kind of fits this general picture that the injury causes problems that mimic many of the features of the human condition, and the repair ameliorates some of these clinical symptoms and problems. This is along the same line of that, and here we show that when we look in the dorsal root ganglia, that there is no loss of nerve cells there, which then points to the suggestion that the changes that we're dealing with happen in the spinal cord. There's a rearrangement of connectivity in the spinal cord that may contribute to development of neuropathic pain. We've also expanded this a bit by developing a visceral pain model, or a model that we can look at certain reflexes that involve the visceral function. So here we're looking at infusing the bladder with saline after a ventral root injury, as you see here. And normally when you have the bladder emptying, there is also a component of abdominal muscles that are activated. And this is also normal in humans during micturition, when we empty the bladder, that there is also activation of stomach muscles. And we can record these, as you can see here. The firings that you see here, these short bursts here are normal activation in rats during a bladder emptying there. That's the way it normally looks. But when we study this also after injury and repair, even though this may be a bit of a complicated slide, the point here is that the activation that we see in the animals in control situation is changed to a more of a upregulated firing after the injury, and over time you have a decrease of that painful-like response. So again, in this model, the visceral pain that is suggested to be coming up after the injury is ameliorated also and shortened in duration when we do the root repair. So again, the root repair is again a potential positive and good thing. So in addition to the complete and both-sided injuries that I've been talking about, we have also recently looked at incomplete injuries, because the reality in the community of people with these types of injuries is that most people are incomplete with degree to lesion. So what we did here was to do a unilateral or a one-sided lesion of the roots, with the opposite side being intact. And this is an interesting lesion because the animals don't need any particular bladder care after the injury, and they are perfectly fine. They can empty the bladder. They don't need to be expressed, as we say, equivalent to humans being catheterized and so forth. And also in humans, if there are lesions only on one side of the spinal cord like this to the roots, typically humans are also able to maintain a good bladder function. But what we did here was, after that lesion, to do the urodynamic recordings, and we found that even though the animals, when they are awake, that they are doing well as far as the emptying, the reflexes are still not normal. And so we have here changes that we can study and outcome measures that the completeness of emptying is affected. Also the firing of the sphincter, as well as the pressure relationships, they're also affected by this injury. And as an additional step, we looked at the pharmacological intervention, and this is the work of Harriet Chang in this model to look at the effect of something called 8-hydroxy-DPAT, which is an agonist for a serotonergic receptor called 1A. And this particular drug acts on receptors that normally are acted on by a brain chemical or spinal cord chemical called serotonin. So what we were able to find here is that the voiding efficiency, the injury-induced problem that we were finding after the injury alone, could be reversed with this drug intervention. So this is actually really encouraging that there is a pharmacological approach that maybe can be used in combination with, let's say, our surgical repair to get that extra push, or maybe instead of a surgical repair in incomplete injuries. So at least it shows to some potential mechanism that may be used as a combination therapy for these types of injuries. So when it comes to the surgical repair of conus and Cauda Equina injuries, we conclude that the surgical replantation of roots can be neuroprotective, meaning that it increases the survival of both motor neurons and the autonomic neurons. We also have shown that they stimulate the growth of axons from the central nervous system to the peripheral nervous system, which is a huge aspect of this, and really critical for anything that later comes with the functional reinnervation. And we were able to show that also bladder function can at least partially return. And we're hopeful that by combining additional approaches, we can get even better results on this. The aspect of sensory effects is intriguing and interesting, because this is one of the first examples of a motor injury having sensory consequences and pain. Most of the models that are used in pain research, almost all models that are leading towards drug development and so forth, involve direct injury to sensory nerves and sensory systems. So this is a different way of thinking about this, that it may unmask some new mechanisms behind pain. And we're also looking at this from the gene expression perspective to see which pathways that may be involved in this model. We also have shown that the end organ is better protected, and that we can also modulate and restore many aspects of spinal cord circuitry, at least at the nerve transmitter level. So these are all bits and pieces that are, positive effects of a surgical replantation after an acute injury. And so some of these may even in isolation be significant as far as potentially being able to reverse injury effect and improve quality of life for people with these kinds of injury. And the interesting aspect also with this approach is that in its purest form, by just surgical replantation, the procedure only uses the person's own tissues. There is nothing foreign. There's nothing from outside of the body. It's a surgical procedure. So it doesn't need FDA approval as far as moving forward towards clinical trials and so forth. It's actually a surgical procedure that, of course, it needs the IRB and other components, but it is not like a device. It's not a drug. It's not something artificial that you bring into a repair and therefore would need separate reviews and so forth. So it is actually, from that perspective, attractive and may potentially be something that can be combined with surgeries that already are taking place. As many of you know from first-hand experiences, is that after the trauma, a lot of the times after a spinal cord injury, after coming into the hospitals in those first few days, that patients are taken to the operating room to stabilize the spine, remove bone fragments or blood clots or things that could put pressure on the cords. It's a decompression surgery. But when those surgeries are taking place for patients with these kinds of injuries, that's all that's really done. It's decompression and spine stabilization, but there is no repair of any nervous tissues. So here is a window of opportunity that what if we could combine, in select cases, this type of approach and possibly have a recovery of function. Of course, there are many other steps along the way to get to that point, but that's kind of the way we are thinking, that the experimental work that I'm describing here is translatable in that direction, and we are actually working in the various steps to get to that point. And of course, this is a team sport, and so we have wonderful people in the lab and collaborators. Much of the work I was talking about today has been the work of Harriet Chang, also former members of the lab, including Alison Bigby and Tao Huang, and also a relative new member, Arthur Yamin, in the lab, and also many of our collaborators, literally around the world. And we are very, very fortunate and that we have had also great research support for these projects, both from philanthropy and NIH and foundations, especially for the early rodent work. We are currently also funded by the Department of Defense for translate part of these steps in the non-human primate model, and we are also looking at expanding our interactions with clinical colleagues and basic scientists that are interested in this approach. So thank you very much.